All right. Welcome, everybody. So excited for this webinar. My name is Sarah Fisher. I'm a Community Engagement Manager on the Cancer Genomics Cloud from Cera. And I will be presenting a little bit of an introduction to this webinar. And then I will turn things over to my colleague, Boris Majić, who is a data analyst at Cera, has been with the company as a data analyst for many, many years. Boris holds a bachelor's degree in signal process, or a bachelor's degree in electrical engineering and a master's degree in signal processing, and is going to tell us all about the wizardry behind MCMICRO and how to use it on the Cancer Genomics Cloud. I am going to, a little bit about me, I suppose, would be appropriate to tell you. I have a PhD in evolutionary biology from the University of Connecticut, and I have been with Cera for just about a year now. My role is in supporting researchers who are using the Cancer Genomics Cloud in their research. But without further ado, let's talk a little bit about MCMICRO. I'm really excited to get to share this information about this tool with you guys. I, like many biologists, love looking at beautiful fluorescent microscopy like this. But the work that goes into actually analyzing these like whole tissue, whole slide imaging files is a lot more than many of us, I think, appreciate. So what is MCMICRO and how does it help us understand what we're seeing in these gorgeous slides? MCMICRO is a scalable modular image processing pipeline for multiplexed tissue imaging. And when we're talking about multiplexed tissue imaging, the multi that we're plexing is many different markers or targets or probes that we are actually able to image on the microscopes themselves. So for example, in fluorescent microscopy, different fluorescent stains would be different targets for the multiplex tissue imaging. So MCMICRO is an automated pipeline for processing these images, and the CWL version was developed in collaboration with HTAN, the Human Tumor Atlas Network community. So this really strengthens our collaboration with this dedicated group of researchers. The workflow allows users to transform these whole slide images into single cell data that they can then use in further downstream analyses. And without having something like MCMICRO, the conversion between images and single cell data requires really intensive computational knowledge, which is a big ask for somebody to be able to do the microscopy and have lots of bioinformatics background. So what about people who have the microscopy but don't have all that bioinformatics expertise? The CGC implementation of MCMICRO allows this point-and-click interface for users without bioinformatics expertise. Users can upload raw images along with channel marker files that describe the multiple targets, probes, or markers that are present in the data. And then they can select which tools they like to run using optional flags. And Boris is probably going to talk more about that. Once the analysis is complete, several outputs are generated, including segmentation masks, counts tables, and really beautiful visualizations. The integration is beyond valuable for researchers and clinicians who process maybe dozens or hundreds whole slided images and want to be able to draw really objective, non-biased conclusions from this data. So there are two main ways to access MCMICRO on the CGC. One way is through the public projects gallery. Public projects come preloaded with input and output files, pre-run tasks, and recommended settings. And in addition, in the public projects, there are detailed descriptions that provide information on what the workflow does and how to use the pipeline. The second way of accessing the workflow is by going through our public apps gallery. So you just click that option on the top blue banner, and then you simply search for MCMICRO in the search bar. And then finally, you select run or copy to move the workflow into your project space. But keep in mind, if you do this, that when you access the workflow through the public apps gallery, it doesn't come preloaded with files. It's just the workflow itself. And this is just sort of a sneak peek. Once your analysis is completed, there are several different output files that are generated. One of them is semantic segmentation. The semantic segmentation in MCMICRO produces 32-bit masks, which are used to quantify pixel intensity, which often equates to protein expression in most images. That's quantifies pixel intensity on multiplexed whole slide images for cytoplasm and nuclei. And then you get a resulting spatial feature table, which is exported as a comma-separated value file for subsequent data analysis. In addition to these tables, though, publication-ready plots are also generated. So on the right side of this slide, you see heat maps and UMAP plots for cluster visualization. You can visit the MCMICRO website for lots of information about the types of files that are generated, or check out the paper listed along the bottom of the slide, and I will be popping those links into the chat after Boris takes over with his demo. Now, it's important to note that things like this don't happen without lots and lots of contributors. So we made this credit crawl so that we could make sure to thank all of the many collaborators that helped bring MCMICRO to the Cancer Genomics Cloud. Thank you to each and every one of you for your efforts on this collaboration. And then also, as a bit of a shameless plug, I would like to remind everybody that we hold office hours every Tuesday at 10 a.m. and Thursday at 2 p.m. Eastern. This is a really awesome community time that we're building. Rawan and I are both available during office hours. It's a time where you can come and ask questions or chat about your research, but it's also a time to come and talk with other people who are using the Cancer Genomics Cloud and share ideas. I know a couple of people here have definitely been to office hours and probably have gotten a lot out of it. I hope they have. But office hours is a pretty chill hang, and you should come. And with that, I will say thank you, and I will turn it over to Boris.
Oh, thank you, Cera. Let me just share my screen and start the presentation. Thank you for the great introduction. It was a joy working with people from both from HTAN and from Harvard University's laboratory of system physiology. I'm sorry if of systems pharmacology. Pardon. It was great collaborating with both developers and users of MCMICRO to bring this tool, which is otherwise a workflow made for Nextflow, which is not a native language for workflows on Cera platforms. So what we actually brought here is we translated MCMICRO from Nextflow workflow language to common workflow language, which is currently better supported on Cera platforms. But soon, and I don't know if I'm spoiling it or not, but soon we will also have a good Nextflow support on all of our Cera platforms. So Cera already gave a great description of what MCMICRO is. It is important to highlight that this is an end-to-end microscopy imaging processing pipeline, which means that you can start with raw data and finish with visualizations and quantifications, and basically, as Cera said, publication ready graphs. Now, before I demonstrate how to use this tool, this workflow on CGC, I will just briefly go through each of the steps that are covered in this workflow. And just to mention that this is happening right now, but the day after tomorrow, Cera will have another meeting with the developers of MCMICRO and HTAN team to publish a newer version of this workflow. So the version which is currently available is a version from a couple of months ago, and in the meantime, we have done a lot of additional work to make more features available in this common workflow language version of MCMICRO. And if any of you want to play with the newer version, Cera or Zelia or Rowan can give you my contact details, and I can give you the new version a bit in advance. So we are expecting to publish it somewhere next week, but if you are impatient, we can let you in on it a bit sooner. So this is the whole pipeline. It is a bit different than the one in Cera's slide because some new features were added, and now I will just briefly go over each of these steps. So MCMICRO starts with illumination correction. This is an optional step, which for you who are not necessarily proficient in image processing, it basically corrects for some defects, or not necessarily defects, but effects of camera. Cameras which are used for microscopy are very sensitive cameras, both to temperature. Maybe you're familiar with the fact that these cameras are cooled to -20 or even -80 degrees Celsius. I'm not sure how to translate it to Fahrenheit. Sorry for my European scales. But these effects can the effects which exist from temperature being higher than 0 Kelvin manifest on our images and can impact the final results. So MCMICRO starts with illumination correction, which generates these dark field and flat field profiles, which carry. I'm not now trying to give a lecture on cameras. If you're interested, we can talk about this later. Or you can contact the authors of MCMICRO. But basically, we can use these profiles to correct for some abnormalities or in our final images. After we have done illumination correction, what neither Cera nor I have mentioned so far is that MCMICRO can work with two types of microscopy images. It can work with whole slide imaging, and it can work with tissue microarrays. This part is suited for tissue microarrays, where you have one object, for example, either a tissue or a bunch of cells, photographed in such a way that you do not fit the whole object into one slide. So you need to create this mosaic image from a multiple starting images. So in this example, we are merging several images to create this mosaic image of two different wells in our plate, which we have imaged. So we have two tissue samples here in total, but in order to take an image of these two slides, we have taken multiple different images. In this case, I believe there were 8 images. I'm not showing them all here. Ashlar, which is the second step of MCMICRO, will perform slide alignment and create one image, which is then analyzed in downstream steps. Another optional step is background subtraction. In some cases, in addition to correcting for dark field and flat field profiles, you might want to perform some sort of a background subtraction. This step requires that one of the channels in your images is marked as a background channel. So removing background requires additional preparation of channel marker file, where you will highlight the background channels. All of this is described in the MCMICRO description in the description of the workflow on CGC, or you can find the same information on the MCMICRO homepage. I forgot to change my slide names. Never mind. But now we are not talking about background subtraction anymore. Now we are talking about segmentation. And MCMICRO can use 5 different segmentation algorithms. Later down the line, any additional downstream analysis is performed separately on each of the different segmentation outputs. I'm not going to go deep into these segmentation algorithms, but on these slides you have a list of different algorithms that can be used with MCMICRO. Each of these is optional, and in the workflow itself, you can choose which of these algorithms you want to run. It might not make sense to use all of these. These segmentation algorithms have different output formats. Some of these will create probability maps. Others would create segmentation masks. And in order to make the further downstream analysis possible, all of the probability maps are converted into segmentation maps. This is the inner segmentation workflow of MCMICRO. It can also perform resizing of input images to enable a better performance of the segmentation algorithms, because some of the segmentation algorithms use machine learning models which are trained on certain size of cells. So if your cell is not the appropriate number of pixels wide, if it is 10 times, if you have a larger zoom than what was the zoom of the model which the algorithm was trained on, you might have some problems and you might want to resize the images. This information can as well be found in the workflow description. Next step in the workflow is quantification, where now after the segmentation has discovered which cells are present in the image, so the segmentation is cell-based segmentation. Now we can quantify the information for each of these cells, and this information is then used in the downstream analysis. Cera already showed some of these plots. But there are 4 different tools that can be used, which is a difference from the previous version. In previous version, I believe that we only used one of the downstream analysis tools. Now there is an option to use up to 4 different downstream analysis tools. All of these tools will more or less do the same kind of analysis, just with the different, slightly different pre-processing or clustering algorithms. And the final step is visualization. MCMICRO uses AutoMinerva for visualization, which will allow. I will show these results later. As I agree with Cera absolutely that looking at these colored microscopy images can be really beautiful and a form of art. But I won't now go too deep into these. Basically, AutoMinerva will not only allow you to look at different stacks of channels. So, for example, here in the left image, 3 of the channels are used, and colored in different ways. And AutoMinera will try to detect which combinations of channels can be useful, or you can just look at one channel at a time. It will also make some groups and try to somehow be smart about what combination of channels carry the most information. This is it for the presentation, and now I will move to CGC and show you how you can get MCMICRO to one of your projects. As Cera said, there are two ways to get MCMICRO. One is by going into public projects, and one of the public project is MCMICRO public project, in which you will be able to find not one but two different versions of MCMICRO pipeline. One is the complete MCMICRO pipeline, which is the pipeline that I have presented so far, which starts with illumination correction and ends with the visualization. But if you want to use MCMICRO with HTAN data is already pre-processed. So now pre-processing steps such as illumination correction, background subtraction, and the stitching of tissue microarrays in separate images is necessary. So additional pipeline was translated to CWL, which is made specifically for HTAN data, which starts from the DRA step, which I haven't presented so far. But the DRA step will try to figure out if there are multiple objects in the picture, and it will separate one image into multiple different images, but each image containing one separate object. So each of the objects can be then visualized separately. So one way is to go to public projects, select MCMICRO public project, and from there, I'm not sure if there is another way, maybe Cera can tell me, but there is this information tag next to the project name, from where you can copy the project and name it however you want. From there, you can also select the billing group, and after the project is copied, you will have it present in your project list. Beside, what is? Why do not I have? I'm not sure what's going on.
It just takes a second to. It's fine. Yeah, it takes a second to copy everything.
Yeah. So in this project, it is not only the application, the two workflows, I believe. There is also some collection of exemplar data which can be used. These are examples from MCMICRO's GitHub repository. Selection of imaging data. I believe that one of these is tissue microarray microscopy, and the second one is whole slide microscopy. And there are also two HTAN images. And I'm not sure if yes, there are also examples of how to run these workflows on the given data. So there are two examples of how to run MCMICRO for HTAN workflow, and two examples of how to run the whole MCMICRO pipeline. Let's try to run of these workflows now. Maybe it's actually not the best idea to show this all the versions, since next week we will have a new version with more options. So here I also have the newer version of the workflow in a separate tab.
Sneak peeks.
Where you can select your images and select your channel marker files. I might even want to show you how these channel marker files look like. We have worked with HTAN team to also bring channel marker files into their data set, as these files were not publicly available before this collaboration. But now you can also browse for channel marker files on HTAN. It basically says which channel contains which sort of information. And this is a simple example of only 8 channels, but your data can have much more channels. It is important that every channel present in the input image is described in the channel marker file. That being said, some of the segmentation, and this is all described in the workflow description, which is pretty detailed, and also contains some benchmarking information. So you can appropriately select computation parameters for your use case, and be able to estimate the total cost of the pipeline execution. I don't think that I should go into each of these options. It might be important to demonstrate that each of the segmentation algorithms is optional, as well as each of the downstream analysis steps. So you can select a combination of segmentation algorithms that you prefer. Some of these are sometimes more problematic than the rest, and you should make sure that your image is the proper size, as you described in the beginning, because if your cells are too small or too large, segmentation algorithms won't detect any cells, and then the workflow can fail, because for the downstream analysis, it is necessary to have at least 20 or 30 cells in your image to be able to perform a cluster analysis, for example. What else did I want to demonstrate? Now we can look at how the outputs look like of one of these tasks. I don't know. Let's take one of the. Oh, this is very big. Never mind. This is an example of a large file. So this imaging file is 7 gigabytes, and it is on the larger side. We have tested the MCMICRO workflows on imaging files up to, I believe, 100 gigabytes. And I have lost myself a little bit. And the workflow will generate 2 outputs. One is the visualizations, which are archived, and then you also have the output directory. Output directory will be present in your project files, and we can look at what is in there. There, in this output directory, you can have intermediary outputs of each of the steps. So, for example, if you're only interested in quantification tables and want to perform your own downstream analysis, you can take one of these quantification outputs. So, as I mentioned before, downstream analysis is performed separately on each of the segmentation algorithms outputs. So for this example, I believe that three segmentation algorithms were run. So I have no idea how to pronounce this, but also I know total of 4 were run here: Mesmer, Cellpose, Elastic, and UNMCIS. And for some of the algorithms which I believe generate probability masks, you can find separate information for nuclei and for the cells as a whole. But for those which produce segmentation masks directly, they do not discriminate between just nuclei and the cell as a whole, so only cell level information is present. So that's the output directory, which contains the intermediary steps, and as well the visualizations generated by this might be the AutoMinerva visualization. So visualizations generated by the downstream analysis. So here there was only one object in our image, so there is only one scene. But if you had multiple objects in your image, you would have multiple scenes here. And depending on the downstream analysis tool, different kind of plots can be generated. And these are the plots that both Cera and I have used in our presentations, where you can see if there are any any clusters of cells that are different from one another. Okay, so that was the output directory. The second output, let's see how big this is. Okay, it's not that big. Is the AutoMinerva visualization. This is a collection of many files, and due to the Valsera and CGC, well, like CGC, not Valsera, due to CGC file structure and the way how we keep files and how we keep track of files, these files are all present on a volume somewhere, and the HTML report generated by AutoMinerva isn't able to track where these files are. It only tracks these files locally. So instead of downloading these hundreds of files, you can just download one archive, which will then contain this HTML report. Then you can open locally, and this is how one of these reports looks like. This is the AutoMinerva report, and AutoMinerva has selected several groups, which are combinations of different channels that you might want to explore, with the description of which channels were used and which color was used to label the corresponding channel. This visualizations here is interactive, and I'm not sure if you can see it, but you also have this scale here, so you can understand at which scale you are looking at. That's it. I was a bit faster than I expected. I'm not sure if I wanted to say anything else than this. Sometimes I talk too fast, but I think it's fine to have a bit more space for questions. Okay, let's finish on one of these pretty images here while we are waiting for questions.
Yes. I'd like to ask one question on the recording, and then I will stop recording so that everybody can feel comfortable asking whatever questions they have. So my main question is, aside from the MCMICRO website and the publications, where's the best place for people to go to get help or advice on like which settings to use and how to make decisions?
Okay, that's a great question. So I'd say that there are two main. They can also contact our support team, which will then probably that question will probably get to me at the end.
Oh yeah, could you show the big blue button? Go to the
oh yeah, of course. So on every CGC page there is this. Maybe not every CGC page.
It's at the very bottom of that if you scroll all the way down.
Oh, okay, yeah. Okay. There is this question mark button which you can use to write directly to our support. You can say that I would say that the best time to contact the support, for example, with respect to MCMICRO, but also with some any other publicly available tools or workflows on the CGC platform, is to already have a project and files which you want to use, and maybe a failed execution where you have tried, so we can see what you were trying to what we were trying to achieve, and then we can be more helpful than if you just contact our support and say, hey, I need help with the MCMICRO without any additional context.
Cool. That's helpful.
That is one way. Another, a lot of information is present in the workflow descriptions. So I would advise that before you start using the MCMICRO for the first time on CGC, you quickly go through the descriptions, understand what are use cases, what are some common issues that you might want to have in mind before you click run and spend some of your resources on a task which is honestly likely to fail if you're using it the first time and you do not really understand what each parameter is for, and it is probably the best way to use CGC. If you're using some apps, try going through the description first. That would be, I guess, the one place that you can find some information. A lot of information that you can use to understand the MCMICRO is to go to the MCMICRO web page. But this web page can help you understand the steps and the process and some parameters. But it is this page is more designed for the Nextflow version of the MCMICRO workflow. So it contains a lot of Nextflow parameters, and there are still some differences between the CWL and the Nextflow version of the pipeline. For example, the Nextflow version, this is something that will probably work in the future to make it even more similar, or maybe not if Seven Bridges, Cera starts and CGC starts the full Nextflow support soon. So this is something that we're currently discussing. So one of the options of the Nextflow version, which is not translated to CWL, is the option to start at any step and finish at any other step, so that not the whole pipeline will be executed. But a lot of information about different options for each of the steps can be found also on the MCMICRO page, and there is also MCMICRO GitHub page where you can find some additional information, or maybe if you want to look at the code and understand how it is. So here you can find the Nextflow code, but perhaps you want to understand. You are familiar with Nextflow, you're proficient with the use of programming languages, you might want to go a bit deeper in these tools. You can visit the MCMICRO GitHub page. All of these links are also present in the tools description. So
fantastic. Yeah, and I'll also share from the chat that one of our collaborators and one of the developers of HTAN of MCMICRO for HTAN, Clarence Yap, says there is a good troubleshooting guide in the MCMICRO website that can help with optimizing parameters, and this is independent of whether Nextflow is being used. So thank you, Clarence.
Absolutely, Clarence and the rest of the team were a tremendous help during this whole process, and there is definitely a lot of information that is present here and on the MCMICRO web page. You can, as Clarence said, there is a troubleshooting section, and importantly, a parameter tuning section for each of the for each of the steps. So.
And I also, just as a sometimes somewhat clumsy bioinformatician myself, I really want to encourage people to try out with the practice data first, rather than trying to run it on your own data right off the bat. Make sure that you know that it's going to work the way that you want to run it before you try to run it on the 100 gigabyte files that Boris is talking about. I'm going to go ahead and stop recording now and let anybody else ask questions.